All right. Okay, here is the IA for with the research question of the impact of heavy metal contaminated soil on seed germination of crop plants. Do you think it's a good topic, or do you consider this as a research question? What's your reflection on that? Just tell me about the title. Do you think that's the appropriate title? According to the criteria. Too broad.
Yeah.
What do you think you should do?
I also. So it says. So crop plant. What kind of crop? And also like heavy metals. What kind of?
Okay, that's the point. That's the point. Why? Because this is not the research question. This is the title. So the title not necessarily needs to be focused. That's the title of your paper. But once you are making your research question, then your research question should be focused. But as long as your title is your title, and you make a focused research question, you can separate them. And actually your title can be a complete title without being that much focused. Okay, so that's the reason I ask you if you feel that this is okay or not. So the impact of heavy metal contaminated soil on seed germination of crop plants. I think as a title would be okay, because still he or she doesn't talk about the focused research question. And then in the next page you will see the research question which is more relatively focused. Okay, so it says, to what extent does increasing copper concentration in soil affect seed germination of mung beans, with the name of the species, which is scientific name, and it should be in italic, and it should be written completely. Okay, so you cannot write V radiata, you have to write Vigna radiata, complete scientific name, as measured by the total length grown and the fresh weight. So this means that even the variables she's going to measure has been already mentioned in the research questions. I think she could have added the unit somehow if she wanted to make it more focused. Like length based on like centimeter and fresh weight. It's not a very scientific definition saying fresh weight, but maybe he could have said biomass. It's more biological than saying fresh weight. Fresh weight is just something that you can say not scientifically for any reason. Okay, so do you have any idea about what I said? Do you think it's what else you can think about this research question? It's okay.
Yeah, it's so it identifies which type of what kind of plants
Okay. Can you speak a little louder, because I'm recording this, so I can in the video recorded so that I can hear, and in the future I can use it. So you can just speak louder. So you are not in the video, so it's confidential. Don't worry about it. It's just a screen. Okay, so background information. Metals can be found in environment sources including water, land, and atmosphere. Metals can be found in soil that is available. And she's going to make a reference about what she wrote, so that's called like in-text citation. Metal ions can be beneficial, and however there are downsides to metal ions. So this is going to be kind of discussion type when you go up and down about the benefits and the harms by making the reference. The examiner has and definitely she or he is the same examiner, because he has the same technique, methodology for giving. She's not writing too much. She doesn't try to make more comments over writing. She's quite summarized person with low amount of the comments about every separate criteria, and even she hasn't mentioned why she has underlined this, which is a little weird, because if you make a comment, then as an examiner you have to write something and why, what does it mean, and why did you make an underline. So she has made an underline, but she hasn't explained why. So when metal has high concentration, it becomes a heavy metal and causes pollution in plants and soil. Maybe that's the definition of this. Because of this definition, she's concerned about why like this explanation. Alright, releasing heavy metal causes a similar rate of pollution compared to volcanic eruption, and she has a reference for that. Toxicity leads plant to death. This is a good part of the this idea, because it shows that once you are bringing an idea, then you find a reference for that. And here is the reference: the toxicity can lead plants to death by causing disorders such as low biomass accumulation and so on. So this is going to be a reference by Singh et al. And also the effect of the volcanic eruption, again by Ali et al. So this means, you know, et al. means the rest of the publishers, but you have to write in this way, et al. Et, I think, is the Roman, probably means and al means all. So mean and all, meaning the rest of the people, the rest of the scientists who are named in this publication. Toxicity can damage human functioning of organs, and this includes the brain, kidney, lungs, liver, and blood, and causes disease such as cancer. Okay, so she has tried to bring some background information which explains very good about the reason that she brainstormed this idea. The toxicity of the heavy metal level increases as more metal waste is added into nature. And again, the same thing among different metal. Copper sulfate is often used, and copper is one of the prevalent of the essential elements for the lives of organisms, and is considered as micronutrient for growth of crop plants. So according to this background, applying low amount of copper sulfate to the soil adds nutrients to the soil to prevent chlorosis. and which causes lack of chlorophyll. This is also has been mentioned or referred to another paper by Allen. So this means that the copper has effect over the chlorophyll production. However, it might cause adverse effect when high concentration is used, since copper sulfate crystals easily kill roots of the plant only in short-terms. So because copper sulfate can kill the plant, may have an opposite effect. So to me, this background information is really nicely done, nicely written. What do you think? So is that the background completely related to the research question? So these are all that you have to be careful and consider when you are writing a step by step and the relation with your research question. So what is the hypothesis? Hypothesis is that increasing concentration of copper metal in the soil of germinating mung beans would cause a decrease in the growth of germination in mung. So this is what she hypothesized, what she expected. The opposite effect of the contamination of the copper metal over the growth of the mung beans. As the concentration of copper ions available in the soil increases, it may be considered as heavy metals. So exceeding the tolerance level of heavy metal concentration would damage the growth of mung beans, since the hard metal might be toxic, causing cellular damage. This is also has been mentioned over a reference about this, that how a toxic metal can be damaging the mung beans growth. Thus, the mass of mung beans and the length grown would be decreased above a certain concentration of copper sulfate. He is going to find out, is there any concentration of the copper sulfate that can affect the growth of the mung beans. Environmental implications: having a high level of pollutant in soil is a serious problem. And again, explanation over the background. Soil pollution is a challenging problem in China. I think she's trying to put more engagement part that she has chosen this topic with the research question. This standard is given for soil protection, but in the sampling areas in China. Because she has done this in China, she needs to go through the Chinese information and the data that they have. So about 94% of soil has rated exceeded grade 2 with high cadmium, manganese, copper, and nickel, about the chemical substances which soil are considered as polluted. So this is also some information about what percentage of this type of chemicals can be considered as pollutants, or the soil is polluted. Soil pollution is caused by human activity and it is a huge environmental awareness how increasing human activity can lead to soil pollution. Therefore, I made an investigation of applying heavy metal substance to plants to evaluate the difference in results of plant growth when different levels of heavy metal are added, to find out the level that plant can afford. And with this result of investigation, I could make a prediction for how much metal can be accommodated in the actual society and be prepared for future pollution. So the variables and the planning until now was the context, and I think she has written a very nicely content with reference to and the connection between the content and the research question. Everything is nicely written. What are the independent variables, like the concentration of the copper metal? So this means that by changing this concentration of this copper, you can see that what will be changed after concentration, after changing the copper. Like the dependent variable will be changed. What are the dependent variable? Are the growth of the mung beans and the fresh weight of mung beans after germination. So two variable has been considered to be dependent, and as you can see how she already tried to put plus and minus. This means that error. One of the criteria in your rubric is talking about the errors, how to show the errors and the deviations. So this is important. This way writing is very important, that you get a full grade or full part of that. Fresh weight of mung beans after germination for 36 hours as measured in grams. This is how she did this measurement. Control variables. These control variables are those who needs to be kept fixed during the experiment, including the incubation period. For all the experiment needs to be done like 96 hours, or the temperature of the thermal regulator to keep it constant, the amount of the water, the watering time she has done at 1 p.m. and the initial mass and the condition of mung beans in the initial state. So all of them are something that she has already controlled in her experiment, and also mass of soil and the soil type. The materials she used: copper, mung beans, distilled water, soil, measuring tape, plastics, plastic pipettes, thermoregulator, cylinders, balance, weighing gauge, beaker, and so on. And then the method that she used by preparing the stock solution using the electronic balance, measuring amount of the sulfate, copper sulfate, and then transferring into the beaker, and then fill up the graduate cylinder until the volume is reaching to the 50, and then the follow-up the steps to fill up 150 ml of the beaker, and then stirring the solution until the copper sulfate appears evenly colored. So this is the way that we prepare the solution, different solutions of the copper sulfate in order to add to the water, add to the soil. For sampling strategy, using an electronic balance, measure the mass and make sure the mass of mung beans is identical before germination starts. So try to pick up the almost similar types of mung beans in order to make sure that they are the same condition. 5 mung beans are placed in separate plastic cups that contain 10 grams of soil. So here is picking up the mung beans and then putting for growth or germination. And the method for preparing seeds to germinate: turning on the thermoregulator at 30 degrees, and then the plastic cup, and they allocate 10 grams of them, and then on the soil of each cup place the seeds with the sampling strategy. Then and then at the end move on the plates in the thermoregulator into a thermal regulator set at 30 degrees. Make sure water is being seated at the time of 1 o'clock, and then checking everything is being under control. So the methods for collection of the quantitative results: after 4 days of incubation, taking out the seeds and washing out the soil and plants, and then dry moisture and plant with the tissue, and then measure the length of the stem using the ruler. This is for measurement of the size of the growth. and then for measuring the mass of the seeds with the scale or the balance. So two variable is being measured, the size and the weight. So risk assessment and ethical consideration, as you can see, she has tried to write as much as possible for these two. Risk assessments such as washing hands and always being careful about using glass Ds. Be careful of not to be hot when taking the glass out of the incubator and so on, and the ethical issues that purposely adding chemical substance in a plant which might count as unethical, but the amount of the soil used in investigation would be thrown to the environment with heavy metal mixture which could be unethical. So the mung beans with the given mixture of water might be polluted caused by the concentration of the heavy metal, and this might be the waste of the food resources. And to minimize the environmental risk, this concentration of mixture would not be made over 10% toxicity. So because of that environmental issue, she hasn't made so much toxicity for her experiment, and which is could be beneficial for that purpose. I mean, not make more toxic soil. Result and analysis. So until here we have already covered the rubrics until the planning, at the end of planning, and then now we are seeing about the results and analysis. So there are qualitative and quantitative data. So the qualitative data is that the sampling method, you can see that how she has set up the different pots in order to grow mung beans. So this is actually very important when you are setting up an experiment, and this is really amazing way of visualization. So how she took the pictures and then showed the different size and the growth of them within different concentration. So this way of doing by showing a very good visualization by taking pictures is really good. So this is a 0.5% concentration of the copper sulfate. This is 1%, 2%, 5%, 10%, and then the bean with chlorosis showing dry bean with mold on it. This is one of the errors of the experiment due to growth of mold on the beans, because this experiment actually should be done under a clean bench, and we don't have a clean bench at the school. This is kind of at the level of university type of experiment, that you need a clean bench which is filtered 24 hours, and you don't get moldy, you don't get any pollution with the bacteria or fungi. Since we don't have it, we try to do it as much as possible with a clean way, and but it's unavoidable. I mean, getting the fungal in your data, in your experiment, will be unavoidable. So the quantitative, as you see, the important part of writing in the table, the title of the table, and the number of the trials for each growth, and the length of the mung beans, the trials. So total length of mung beans grown after 4 days of incubation in different concentration of copper sulfate. So this is this table showing the length, this table showing the mass of mung beans or biomass. And important thing is how she has tried to show the errors, plus minus. This is important. Number of the trials is 5. This is also another important for doing experiment. and she has done two variables, and you can see one is for length, this table, and the other one for weight. And how she has done the data processing, I think the examiner just made a comment over here, what was the initial mass. So she didn't mention the initial mass. It's better to mention that part as well. The data processing, average total length of and mass of mung beans after 4 days of incubation in different concentration of copper sulfate. So the average length was this. By increasing the amount of the concentration of the copper sulfate, you see from 0 to half, 1, 2, 5, and 10. And then the average length of mung beans, you see that how is a strong correlation by decreasing the average length of the mung beans. And standard deviation showing in the table based on the average and the standard deviation, and the graph, a scattered graph of the total length of mung beans growth after 4 days incubation and different concentration of the copper sulfate. So actually she has tried to show with one trial, and each trial has been made a line over the graph, and it's very clear that by increasing the amount of the copper sulfate, then we will have the less length of the mung beans. So by reaching the 10, then the amount of the length will be showing decreasingly, and it is consistently in 5 trials. All of them show the same thing. So if you want to show the error bar in one graph, this is how you show that. And as I told you before, how to make an error bar is very important, that you are using the appropriate method. So you see that, for example, at this area, you have the bar is longer or wider than the bar here, than the bar here, and at this stage, this is because there wasn't that much deviations from the mean, or standard deviation was not high. So that's why you get the error bar, and this is the line of the best fit. And the R square, as you can see, is 0.8, which is a very strong R. It's completely supporting the correlation. However, if you want to make sure, or if you want to do it more appropriately, you can also do the Pearson correlation and bring more statistical analysis. If you want to do it more, it's okay, but here 0.8 is completely strong, showing the strong correlation. So average length of mung beans over 4 days of incubation is shown with plus minus standard deviation. And graph 3 is the scattered graph of mass mung beans growth after 4 days. So the next things to measure was biomass, or the calculating the mass after increasing the amount of the copper sulfate, and also it has the same pattern. She has drawn different lines for different trials, but all of them are following the same pattern. By increasing the copper sulfate, the mass also will decrease, and here is the error bar for that. So and you can see that the R square here is also 0.9, and that's another strong correlation showing that by increasing the copper sulfate, the weight also will decrease. Statistical analysis. This is something that if you want to compare populations but more than two, then you have to do ANOVA. As you see here, to further validate the results, statistical ANOVA is chosen to be performed, and you see that the ANOVA is validating the significant difference in the length among the means of 6 different concentrations of the copper sulfate. So she wanted to show that if the different concentrations of the copper sulfate will show the significant difference in terms of the weight or their size or their length. So in order to do that, the ANOVA, the source of variation between groups of concentration and within group of concentration, you can do this ANOVA just by using your calculator or the software or even online calculation. So and it gives you the significance, and finally you can evaluate like this, because the p-value from the table suggests that there is a significant difference in at least one pair of the means, and the p-value is given at 4. It is less than 0.05. This means that the null hypothesis is rejected. So when the p-value is less than 0.05, which is the percentage that we are looking for, or like saying that in 95 confidential level, then it is less than 0.05, which is significantly different, and this means that there are differences in terms of their length. So each copper sulfate had effect over the average of the length, and each length showed the differences between significant differences between each amount of the copper sulfate. And the same analysis, the same way for mung beans. This is really good example of how to use the ANOVA, because if you want to compare more than 2 populations or more than 2 data, so you should use ANOVA. However, ANOVA, if you want that ANOVA shows which 2 populations having a significant difference between each other, then you have to use the ANOVA that will show that 2 by 2 as well. Usually the online ANOVA calculators doesn't show that. And if you want to get that information, you have to go through finding the ANOVA statistical analysis that shows all the 2 by 2 significant differences. Okay, so scientific analysis. Throughout the investigation, I found the concentration increases the drier the beans have got, therefore leading to a decrease in size and mass of the bean caused by low biomass accumulated. I think she has tried to interpret what she has already got with the data, and it is obvious everything. By increasing the copper sulfate, the length will decrease or the mass will decrease. Okay, so conclusion. As supported by the trend lines shown in graph 2 and 4, the research question can be answered that increasing copper concentration in soil decreases the growth and the fresh weight of the mung beans, and this is further supported by the statistical analysis of ANOVA. Okay, and then evaluation. There are several strengths and limitations throughout the investigation. So here is this area is the area for modifications and further research and the application and solution. So I try to decrease the ways that error might occur by selecting and managing 7 controls as written above. Therefore, this could lead to a result of having significant patterns in my data collection. However, there were also limitations throughout my experiments. First, 10 grams of soil is not identical for every trial. Difference in particles of the soil might change the absorption of water, which might affect the bins to absorb the concentration. The location of water supply might not be always consistent. So she's trying to show that the way that can be done better, or the error that has been happened during the experiment, and the modification and further research, that when investigation is done, again the improvement made to develop my experiment, I would eliminate those weaknesses by increasing control. So the way that she's offering, or she's suggesting, is that by increasing the control variables for higher credibility and reduce the human errors. But I think this modification and further research is too much wide. It's not very focused. What do you think? Because you can say this to every type of modification and further research. Anyway, reducing human error, that's very common. Application and solution. Copper sulfate is created naturally in the plants to defend themselves, working as a pesticide. Furthermore, it can foster the growth rates in plants with an effect of fertilizer. Therefore, it is natural to find heavy metal in soil sampling, including China, where it suffers with high heavy metal concentration levels. However, since China has shown a high rate of concentration level caused by human activities, increasing the rate of concentration increases the awareness of environmental issues. This could result in the result of killing the roots of plants and decreasing the quality of vegetables. It's a little vague here, that's why the examiner made a question mark over here. Therefore, it is important to find the level of concentration so that the soil is not at polluted level, which harms the plants. Okay. So, what is your idea? You want to go through the rubric and compare with the examiner, but before that, what do you want to say? Do you have anything to say? Not really? I think I reviewed everything by the way that I explained. So even you didn't read it, you could, just by listening, you could get the idea. Okay, so I want to compare. So I hit here with my compare my analysis and my grading with the examiner grading. That would be a perfect practice. So, the first criteria, identifying the context. So, about the student's report, state a relevant research question, state a relevant, coherent, and focused research question. So I gave a state a relevant research question. I think both would be okay, but anyway, not a complete 6 can be given to this. However, as you can see the context, you see that the examiner also has given a very high mark, which is 5. You completely agree with me, and I also have given for the context as 5, so exactly the same. Okay, so the context 5, because discusses the relevant environmental issue, either local or global, and provides the context for the research question. That was really nicely explained the connections between environmental issue. So you saw that the context was really good. So what is your grade? Really? Okay. Planning. For the planning, I think I have some deviation with the examiner because I gave 6 because I thought that the planning was perfect and she has done whatever she could do that, but the examiner has given 4, and my judgment is that because the design was repeatable, method appropriate to the research question, allowed the collection of the sufficient relevant data. I think the relevant, so it was sufficient and it was relevant and it is repeatable. I have no idea why the examiner is a little harsh on this part. Justifies the choice of sampling strategy, describes the risk assessment and ethical. This is perfect. Definitely, I agree now with this after reading. Describes the choice of sampling strategy or justifies the choice of sampling strategy. Wasn't right? Yeah, she didn't justify, right? Yeah, I think so. Maybe that's why it could be this at this level. And design a repeatable method appropriate to the research, but the method does not allow for the collection of sufficient relevant data. It could be both in this and that because the germination was completely showing everything. It was really okay. The germination was okay. But anyway, if you want to be a little harsh, which most of the examiners are, they try to, as much as possible, reduce the grade, reduce the mark. So anyway, I gave 6, but the examiner gave 4 for planning. And then next one, results analysis and conclusion. Yeah, you see that the results analysis and conclusion, again I gave 6 because I thought that she has done perfectly the analysis, interpretation, the data, statistical and everything, construct the diagrams, charts, graphs, relevant, quantitative, qualitative, yes, analysis the data correctly, completely relevant patterns are displayed, yes, interpret trends, pattern, relationship in data so that a valid conclusion to the research question is deduced. I think it should be 6. I really don't agree with the examiner at this point. She or he has given 5, and again I have given 6. What was yours?
Probably 5.
5?
Yeah, since 5 and 6 is at the same column, so maybe it depends on
yeah. Your mood, right? But you know that usually it will be given 5 if you have 2 elements from this category and 1 element from like 3 or 4. So in that case, you don't give a 6 fully marked. But if everything is inside the 5 and 6, I think it's okay to give 6. It depends on the examiner. Okay, and the next one, discussion and evaluation. So let's see. We are almost the same with the examiner. So I gave 2, examiner gave 3. Okay, what was my reason? Describes how some aspects of the conclusion are related to the environmental issue or evaluates the conclusion context environmental issue. I think she wasn't completely linked the environmental issue during the evaluation, so that's why the reason. So, and then identifies some strengths and weaknesses and limitation of the method, describes some strengths and weaknesses, Yeah. Suggest modification and further areas of research. I think I was a little harsh on this, and I could give 3 or 4 as well. So now, if I go back and want to regrade, I will give on this either 3 or 4. What was yours?
5.
You get 5. 4 or 5. What did you give? 4. Okay, but the most difficult part, that is very arguable, is that the application. So the examiner has given 0 means no, means the student's report does not reach a standard described by any of the descriptors given below. That's too harsh, right? So I think it states one potential application and solution of the environmental issue that has been discussed in the context, describes some strengths, weaknesses, and limitations. I think she has given some applications, but anyway, I think 1 was okay, but the examiner has given 0. Okay. And then, did you agree with the examiner, or what was yours?
At least 1.
At least 1.
At least 1. Because she has done some. So in that case.
Yeah, okay. Communication, yeah, perfect. So we don't have any argue here. Examiner has given 3, and also I get 3. The report is well structured and organized, more consistent with the appropriate terminology, and the report is logical and coherent. So my total is 23, but the total of the examiner is 20. So I think our major difference that we were not agree with each other was on planning. So I gave a 6 on planning and a 6 on research and analysis, but examiner didn't give the full mark. But still, I think we are almost at the same page. What was your total? What? 17. You are too harsh.
Harsher than examiner.
You are harsher than examiner? Ah, she, yeah. 19. Okay.
22.
22. You are closer to me. All right. Okay, that was the end of this IA.